Okay, hi guys. So today, as Kelly said, I'll be presenting on aging cells, but before I can do that or get into the paper, we have to define some background information first, specifically what a cell looks like normally. So normally the cell is going through a cell cycle, which just means that it's dividing and it's replicating. Oh, okay, it's fine. And a cell isn't always doing this, so different things can pull a cell out from a cell cycle. It could be something like natural aging, because over time cells lose the ability to replicate, or it could be something like natural or unnatural damage, like a miscopy in the DNA that doesn't get repaired, or something like chemotherapy that damages the cell beyond repair and has to leave the cell cycle. So what happens once a cell is out from the cell cycle? Well, two things, either senescence or apoptosis. So senescence is just removal from the cell cycle, but the cells don't die, and apoptosis is removal from the cell cycle and there is cell death. So the main difference is the death part of it. So the researchers knew that senescent cells could avoid apoptosis, but they didn't exactly know how or what makes a senescent cell specifically go into senescence and not apoptosis. So that was the goal of the paper. And something else to keep in mind before we get into the paper is senescence a good or a bad thing? The answer is it depends, but as you get older, it tends to be a bad thing. Senescent cells begin to accumulate with old age, as I said, and as the damage starts to build up. And they have, once there's a lot of them, they have an inflammatory effect called SASP, which just means that they're damaging the tissue around the senescent cell. Those healthy cells around them get damaged, and this can lead to some problems in the immune system, with your neurons, energy level, all things that we associate with aging. So for the purposes of this paper, we're going to think of senescence as a bad thing. Which brings me into my paper. Targeted apoptosis of senescent cells restores tissue homeostasis in response to chemotoxicity and aging, or in simpler terms, forcing aging cells to die in order to restore tissue health. So some background information, or some initial research that they did before going into the paper, they found that senescent cells are primed for apoptosis, but they're stopped somehow. So I think it's easiest to think of this as a factory. So cells are going through this conveyor belt, this process going into apoptosis. They're getting ready as if they were going to undergo apoptosis, but then they're stopped somehow. There's some machinery at the end that pulls the cells off the track of apoptosis and makes them become senescent. But the researchers didn't know what this machinery was, so they wanted to find out what is making the senescent cells senescent. So they looked at levels of mRNA for these different proteins at the bottom, the x-axis. And what they found is that they had increased levels of this red bar, which is FOXO4. And since there are increased levels of mRNA, this suggests that there are increased levels of the protein FOXO4 showing up in the body. So they did some more research, and there have been some initial research actually pointing them towards FOXO4. And they found that FOXO4 does in fact play a role in senescence and inhibiting apoptosis. They didn't exactly know the specifics of that, but they do know that it plays a role. So they wanted to look further into that in the paper, and they came up with an idea. They thought, what if we actually blocked FOXO4? So again, FOXO4 plays a role in leading to senescence and inhibiting apoptosis. So the researchers wondered, what if we were to block FOXO4? What would happen? So I'm going to ask you guys the same questions. If you were to block FOXO4, what would you expect to happen? I know it's kind of confusing because there's two inhibitors, and you could think of two inhibitors as negative, canceling out. So if you were to block FOXO4, what would you expect to happen in terms of apoptosis? Yeah.
You'd see like a decrease in cells staying in senescence and a decrease in cells like proceeding with apoptosis.
Yes, exactly. So they hope that blocking FOXO4 would lead to apoptosis. So they did some research on this to test if this was actually true. They used a method called short hairpin, or RNAi, which is RNA interference, which essentially just blocks the RNA from being expressed, which eventually will lead to the protein being blocked from being expressed. And the red bars are the ones where they blocked FOXO4. And what you can see is that there's a sharp decrease in the senescent cells where they blocked FOXO4. And what they were looking at is percent viability and percent colony density. which in both cases, the lower that percent is, the better, because that means that more cells are dying, which is what they want to happen. So you can see by blocking FOXO4, these cells are dying off, which is good. That's what they expected to happen. It's also important to note that the control cells aren't dying, which is really important because they've never been able to specifically block senescent cells without killing off the healthy cells as well. So this means that this method works pretty effectively. Yes?
Are these in cultured human cells? Or is
No,
this-
so that's a good question. These are actually in vitro, which are just like plates in a lab. So they can't be done in vivo with this method because it's RNA high. So that was the main question that they had of what could they do to be able to do it in vivo, like in a mouse or eventually in a human. So what they did is they created a FOXO4 dry peptide, which is this yellow molecule there, the dots. And the mechanism works like this. So normally P53 is going to play a role in inducing apoptosis. But once FOXO4 is present and active, it's going to bind to P53, so P53 can't do its job. It can't induce apoptosis. But when FOXO4 dry, their peptide that they designed, is present, it binds to P53 instead of FOXO4. This just means that P53 is again free to do its job, and it can induce apoptosis. So I know it's kind of confusing, but in a simpler pathway, again, P53 is going to lead to apoptosis. Once FOXO4 dry is present, it's going to block P53, and once FOXO4 is present, it's going to block P53. Once FOXO4 dry is present, it's going to block FOXO4. So again, those two inhibitors are going to cancel out, and hopefully FOXO4 dry will lead to apoptosis. So they tested this out. They looked at percent viability. Again, you want the number to be lower because that means a higher amount of the cells are dying off, which is the goal. And what they found is that there's a sharp decrease in this green line, which are senescent cells given FOXO4 dry, which is, again, really good. This means that once FOXO4 dry was given to the cells, it can A, kill them off effectively, and B, only kill off the senescent cells. And those other healthy cells are not going to have an effect on them. So this means that FOXO4 dry is effective and selective. Yes?
What's FOXO4L?
That is another type of peptide that doesn't actually block it, just like a control peptide. So they know that they can kill off the senescent cells, but what effects does this have in the body? So take a look at these mice here. The before pictures are just mice with increased levels of senescence, just like old age for mice. And the green column is the same exact mouse taken later once they had FOXO4 dry administered to them. So take a look, see if you can notice anything different in the before and after picture. Anyone have any observations? Yes?
Well, all of their coats are even bigger than they were before.
Exactly. So the researchers noticed this too. They saw that their fur density was improving by a lot. It's kind of hard to tell, but you can see where the green arrows are. Their fur is growing back, which is really cool. This is a cool effect that FOXO4 dry has on the body. And also it's interesting because what do we associate with hair loss? Usually old age. So this is an effect of aging that essentially FOXO4 dry is able to reduce. So that's cool. Another thing that they looked at, take a look at these pictures here. The mock mouse is the same as the before picture on the last slide. It's essentially just an old mouse with increased levels of senescence. The dry mouse is the same type of mouse, but it was given FOXO4 dry. So take a look, and as you can see, the mock mouse, the old mouse, essentially just sits in the corner, doesn't really move around much, but the dry mouse is exploring the cage, it's running around, it's moving a lot more. So they looked more into this, they did some more studies, and they found that the mice given FOXO4 dry, their exploratory behavior, which you can see above would be moving around the cage, exploring their surroundings, that increased by a lot. Their responsiveness also increased, so that would be something like giving them a little shock. So compared to the aged mice, their responsiveness was way better. And then lastly, their general voluntary activity, like running on a wheel or again moving around their cage, also went up. And not only does FOXO4 dry help improve these things, but normally in a mock mouse, as in those aged mice with increased levels of senescence, as you can see, their activity level is pretty close to 0. So FOXO4 dry is not only improving these effects, but it's bringing them back up from a really, really low level. So it's extremely effective. And once again, what do we associate with low levels of activity and low levels of responsiveness? Aging. So another effect of aging that FOXO4 dry is able to help with. And both of these things make sense if we think about it, because as I said at the beginning, increased levels of senescence can lead to effects of aging, and by killing off these senescent cells, they're reducing some of these effects of aging. So the next thing that they looked at is chemotherapy-induced senescent cells, which are just senescent cells that have that unnatural damage like chemotherapy that pulls them out of the cell cycle. So why would they even look at this? Well, in this case, also increased senescence has negative effects. First, chemotherapy right now is not as targeted as it could be, and it ends up targeting these normal cells around the cancer cells. Those normal healthy cells become senescent, and again, increased levels of senescence has that SASP, that inflammatory effect that leads to aging and damages the tissue around it. But also, this means that those senescent cells, since they're not dying, are taking in this toxicity from chemotherapy, which has negative effects on the body. As you can imagine, chemotherapy is pretty harmful. This also means that since the chemotherapy is really spread out, the dose has to be higher, which means that more of those normal cells are going to become senescent and going to take in the toxicity, which means that the dose has to be higher again, and then it just becomes this cycle. So the researchers wondered, first of all, could we even kill off the senescent cells that are induced because of chemotherapy, whereas before they were just looking at naturally old age senescent cells? So can we even kill off the senescent cells in the first place? And if we do, what effects will it have on the body? So to answer the first question, can we even kill off these chemotoxicity-induced senescent cells? They compared senescent cells that are naturally aged, which is the first graph A, to senescent cells that are senescent because of doxorubicin, which is a chemotherapy drug. And they, again, were looking at percent viability, and you want the number to be lower because this means more cell death, which is the goal. So what do you notice about these two graphs? Essentially, they look exactly the same, which means that FOXO4 dry is equally as effective in killing off regular senescent cells or senescent cells that are induced by unnatural damage, like chemotoxicity, which means that FOXO4 dry works in pretty much all types of senescent cells. So this is good. It means that FOXO4 dry is pretty effective. Second question, will it have any other effects on the body? And the question that we asked was, how can they test doxorubicin's effectiveness now that these senescent cells are killed off? A problem is that we realized that these mice didn't actually have cancer in them. They were just giving them doxorubicin, the chemotherapy drug, to see if it would induce senescence and whether or not they could kill off those senescent cells. But since there wasn't actually cancer in them, we can't really test the effectiveness of doxorubicin, the chemotherapy drug, because we can't tell if they're killing off the cancer effectively. So it's a good starting point because they know that FOXO4 dry works well in both types of senescence, but they need to do further research with cancer to see if doxorubicin or any other chemotherapy actually works better with the senescence being killed off. Okay, so some other things that FOXO4 dry does that we didn't get to talk about for time. So in the paper they also mentioned aged mice that were given FOXO4 dry, their body weight increased. So normally aged mice their body weight tends to fall. FOXO4 dry was able to bring that back up to normal. Kidney and liver function, where normally in an aged mouse is really low and poor, once FOXO4 dry was given, it's able to increase and bring it back up to its normal level. And then lastly, frailty was reduced when FOXO4 dry was given, and other strength-related tests, the mice given FOXO4 dry did way better than they would have if they were just aged mice. Yes.
And it's not period are you talking about? How long until you see effects when you add this to the mice?
It depends on the dose given, because this is the first time that they've ever used FOXO4 dry, so a lot of them they were seeing over time the dose. I don't know specifically the time. I can get back to. So these are just some examples that they found that FOXO4 dry can do. these are just the ones that they found also. So again, this is the first time that they've ever been able to target and kill senescence effectively and selectively. So perhaps further research could tell us some more things that Fraxophore dry does, or some more things that killing off senescent cells does. Okay, so what does this all mean? So some things to consider. One, everything else we didn't get to cover today, just for time. They went into a lot of detail about how cells become senescent. I know we just kind of covered that briefly, but they figured out a lot about the protein p53, how they created the peptide, how senescent cells choose senescence over apoptosis. So this is really helpful for further research to be done, because they pretty much unlocked this way to kill off senescent cells, and they know how to do it, and they found out a lot about senescence. So that's really good and really important. Second thing, how likely is it that Fraxophore dry will be used as a drug alongside chemotherapy? Unlikely as of now, because they need to do further testing with actual cancer to see if they can in fact make chemotherapy more effective by killing off senescent cells. They don't know that for sure yet. Also, Fraxophore dry wasn't specifically made for chemotherapy, but hopefully further research will make either a better peptide or figure out if chemotherapy actually is better with Fraxophore dry. And then lastly, what I was thinking a lot during this presentation is oh my gosh, what does this mean? Are we all gonna never age ever again? Probably not. This research especially was just, the focus was mainly on figuring out the basics of senescent cells and could we even kill them off in the first place. But I do think it's important to think about these questions since you saw some of the effects of aging are decreasing, and by killing off senescent cells, that is a main part of the aging process, or senescence is a main part of the aging process. So I do think it's important to ask whether this research is ethical in the first place, and if it is, whether or not further research is ethical, especially with the goal of stopping aging. I have some examples down here, but I'm going to turn it over to you. If you have any points for either side, or you can answer any of the questions whether or not this research is ethical, if further research is ethical, is any research ethical? Okay, yes?
I have a question.
Yeah.
Did they mention anything about lifespan?
They actually did at the end. They said that they kind of justified this paper, or a point that they brought up is that lifespan is projected to increase. I don't know by how much, but it is going to increase. And they said that since the lifespan is increasing, we have to come up with solutions for a health span, which is just the number of healthy years, versus lifespan is just the number of years you're alive.
So their mice actually seem to live longer?
Not they weren't looking at lifespan specifically, but they were looking at the health of their life over time. If that makes sense. So the goal wouldn't really be to increase their life, but to increase the amount of healthy years that they've had in it. Okay, so if anyone has any other points about the ethicalness of this paper or further research, I also have some points on. Okay, so
I actually had another question too. So you mentioned that senescent cells affect the surrounding healthy cells, poorly or negatively affect the tissue. So did the paper go into in absence of those senescent cells, was the surrounding tissue able to regain health, or
I don't think that they looked at that. I don't think it's I mean, it's tricky because they did find that some of those effects are being reversed, but I can't tell you on the cellular level. But they probably did go into detail about that. Okay, I can talk about some of these points at the bottom. So I have some points for each side of whether or not research about aging is even ethical in the first place. So over the weekend, my favorite TV show, Twilight Zone, I was watching an episode in which the man became immortal, which is kind of far from what we're talking about today, but essentially he wasn't going to age anymore. And he was this is all he wanted in his life. He was a hypochondriac. He was always worried about dying and getting older. But as soon as he became immortal, as soon as he lost the ability to age, his life lost all meaning. He no longer had excitement to his life, he no longer had a sense of risk, and this can be taken as like an argument against aging, like research towards stopping aging, because if you're stopping aging, like life does lose its meaning, perhaps. Like there's less excitement. But then on the other side, one of my favorite TED Talks is by a Cambridge biologist, Aubrey de Grey, and he argues two things. One, we need to be doing this research for our future. It's not up to the scientists to decide whether or not it's ethical. We should be doing this research anyway, and then figure out later and let individuals decide what you want to do with it and whether or not to use it. And then he also says it's easy to stand up here and say, like, we should just accept our fate that aging is going to happen and like live while we're young. But he asked the audience, like, how many of us actually want to age? Like, none of us can say that the aging process is enjoyable. Like, those effects that we were talking about, immune deficiency and neuronal problems, no one really wants that. So, it's easy to say it's unethical, but on an individual level, like, many of us would want to stop aging. So, I don't know if you guys have any other, yes?
It could also cause overcrowding and overpopulation over time, because, like, if you for example, Japan, there's like a significantly increased amount of like older elderly people, and then that could lead to like maybe job problems and stuff.
Yeah, so that's another thing. With this increase of lifespan that Shirley was talking about, and more healthy years, like perhaps people are going to work longer, or there's just going to be overpopulation problems. So that is something we should think about when we're either alongside the research or before the research. If anyone else has any other general questions about the paper too.
I actually have another point maybe to think about. Like, I guess we could also think about the fact that if we're like increasing like the healthy lifespan, and we're not like making people immortal, but just increasing like the health, increasing the amount of healthy years someone has. Like, I don't know, maybe like overall like human like progression will be like better, because like people who have the ability to like think and like come up with like inventions, like all this stuff will be like having more years to like discover that and like be in like their right mind and like the right abilities.
Yeah.
In the paper, did they talk about how the FOXO4 drug was going to be administered if it was to become an anti-aging?
Yeah, so I'm pretty sure that they injected it. Yeah, it was pretty much just how was FOXO4 dry administered.
Yeah.
I'm pretty sure that they injected it, but yeah, I think.
This might be outside the scope of the paper, but to the best of your knowledge, are there any neurological disorders that are associated with senescence of neurons that could be addressed by something like this?
I don't know specifically, but I do know that it can lead to some of those problems. I can look into that. Okay. Next week we have Jeremy Newman presenting on rare diseases, so come out next week, I think.
Yep.